Our next speaker is Dr. Keating, and she is a professor in the Department of Animal Science here at Iowa State University. Her research group investigates the mechanisms behind ovotoxin exposures that compromise ovarian function and the ability of the ovary to mount protective response to such exposure. She is the author or co-author of over 90 peer-reviewed articles and 9 book chapters. She currently serves on the board of directors for the Society for the Study of Reproduction. Please join me in welcoming Dr. Keating.
Okay, can you hear me okay at the back? Okay, good stuff. All right, so I'll come out here so I don't get lost behind this thing. You won't be able to see me otherwise. So today I'm going to talk to you about some effects of chemicals on fertility, and I'm going to talk mainly about, in the beginning, some of the general effects of ovotoxicant exposures and how they can affect fertility. But then I'm going to get into how heat stress we believe is an additive effect in pigs. So I'm going to take you for the first while through some of our pig work and try and convince you that they kind of have an additive effect of each other. But then at the end, I'm going to introduce a class of chemicals that are known as perfluorinated chemicals, and you might have seen them in the press recently as forever chemicals. And they're really everywhere in our environment. We really have no idea the level in our swine and across our swine farms, and I think it's really something that we need to be paying attention to. All right. So this is kind of a general schematic of everything that I work on. And I'm not going to tell you about all of it, thankfully, before coffee. But some of the things we've worked on over the years are water contaminants, herbicides, for example, and some of these have been quite controversial, but we've gone ahead and started to do some studies. We work on how components of fossil fuels and plasticizers can have effects on fertility in females. This, of course, is not really relevant to our swine, right? We don't want to put them on chemotherapy for cancer treatments. However, right, those treatments often do end up in our water, and essentially whatever humans take in and then pee out ends up in our water. So illicit drugs for medical reasons, and also drugs that women take for hormone control, birth control, right? They all end up in our water and can have effects on not on our wildlife, on humans, but also on our production animals. And then in the last few years, we've been really working hard on figuring out why there's additive effects in obese humans, right, of chemical exposures. And we've kind of translated this now into heat-stressed animals, because the metabolic changes that happen are essentially the same. So they get high insulin in their floating around in their body, they get a leaky intestine, and we see a lot of the same infertility effects. And so we've been working quite heavily on that. So this is a cartoon of the ovary, and it's essentially pretty much the same regardless of species. There are some species differences, but realistically, it's pretty much the same. And females are born with this set number of oocytes or eggs. And once they run out, you're done, right? In women, we know this as menopause. They get activated to grow in response to different hormones in the body, right? And eventually you have ovulation, and hopefully fertilization will occur. And then this structure forms what's known as the CL, or the corpus luteum, right? And so there's lots of structures that are developing kind of all the time within the ovary, and any of those can be targets for chemicals. And so this is kind of some of the phenotypes or some of the effects of what we term ovotoxicity or ovarian toxicity. We can get loss of these structures, complete death of the eggs, essentially, and a female will become infertile. Her ovary will be cleared out. Not only is she lacking now eggs, she's also lacking the proper hormonal environment that they need for the immune system and other health effects. We get DNA damage, and that can be passed on to the next generation. That's a concern. We get problems with ovulation and infertility. Many of you know that you often have problems breeding your animals, and why is that, right? You lose a lot of money in that. Every day costs money. You can have hormone disruption. Pregnancy loss, especially early embryonic loss, is a concern for production animals. And then impaired reproductive behavior. That's all under control of the hormones in an animal's body. So if you can't tell an animal is in heat, that can be an issue, right? Or they might act like they're in heat when they're not. So all of these are effects of what chemicals do on the ovary. So this is kind of what I'm going to tell you about today. Some of our work on how endocrine disruption or hormone disruption happens because of chemical exposures, how the ovary alters making hormones, changes to proteins within the ovary, and then how the ovary changes how it can basically detoxify chemicals. And so I'm going to mainly talk about this in pigs and introduce heat stress pigs and why that's kind of an additive almost like an additive toxic exposure. But then I'm going to tell you just at the very end about a study we've done, not in obese female humans, but in obese mice. And I'm telling you this just to make the point that there are differences in the effect of a chemical exposure. And I want you to think about the fact that if this is happening in our animals, whether they have a higher body mass or if because they're heat stressed, I want you to kind of keep that in your head. All right. So I kind of touched on this already, but this is my really bad cartoon of an ovary to show you why I'm a scientist and not an artist. But these little purple circles here, these represent the very most immature oocytes, or the egg store in a female. And then they grow and they start to get bigger, and you get these extra cells around them, and then the action of those cells are what makes estrogens, right? And that's the primary hormone required for female health. It's also the hormone that's required for animals to show that they're in estrus. After ovulation, then hopefully you have a CL that's formed, or many CLs in litter bearers, and that's what produces progesterone. So two very important structures within the ovary that produce hormones. And in a pig, a pregnant pig, if the CL is destroyed at any stage of her pregnancy, she will abort or she'll deliver, right? So pigs especially are very dependent on the CL. So if these are destroyed, you're going to have problems with fertility outcomes, right? Because they're not going to ovulate the oocyte. You're also going to lose production of these important hormones. But if these more immature follicles or oocytes are destroyed, the animal's going to be infertile. There's no coming back, right? You can't replenish them. That's it. They're out. And so it's kind of like you take all your money out of the ATM, right? Unless you've some wonder, right, or some miracle happens, it's not going to come back. And so this can result in all of these different effects. Really depend on the structure within the ovary that's targeted, and chemicals can destroy lots of different structures. I wish it was as easy to say that we know that this is how every chemical works, but it isn't. They're all different, and so we need to figure out what they do. So we know that our liver is important for detoxification, right? And you'll hear people say they're going on a detox diet and all this, right? It drives me crazy, because your liver really is what detoxifies most chemicals that come into your body, as well as endogenous chemicals in your body. But metabolites can be formed within the liver. They can go off to the ovary. The parent compound can also go to the ovary. Almost all your tissues around your body can detoxify chemicals to which they're exposed. You're not just relying on the liver. And so we know the ovary can do this, and we've been working on this for several years. And so the ovary can defend itself by biotransforming or changing those chemical structures to make them more excretable from the body. They can fix DNA damage. The ovary has the ability to do this. And actually, I think follicle death is, so loss of these structures, I think that actually is protective. If that oocyte, that critical, egg is damaged, I think that's a protective mechanism to get rid of the bad stuff. So we know that the ovary has the capacity to do this, and we know that it's important. I'll start first with the work on the heat stress and kind of try and convince you that it's an endocrine disruptor. And so what that means is that heat stress itself, from all the work that's been done at Iowa State, not only by myself, by many other groups, including Dr. Gabler, who's going to give a talk at the end of this session, heat stress causes changes to the body. Insulin becomes very high despite the fact that the animals go off feed. They get high levels of circulating prolactin. They get a leaky gut, and they suffer what we know as seasonal infertility, right? It's getting warmer. We know this. We're having more extreme weather events, and so climate change and heat stress, especially for animals, is a concern. So we showed years ago, this is from 2015 now. It seems like yesterday, but it's not, right? It's a while back. So this graph here, what I'm showing you, the white bars are pigs that were in thermoneutral conditions, and the black are heat-stressed pigs. And this is proteins in the ovary, and these are the mRNA encoding those proteins. And these ones in particular, these last two are very important. STAR takes up cholesterol by the ovary and converts it to estrogen, and CYP19A does that last step, right? So they're important for making estrogen within the ovary. So we know in heat stress that they're altered in the pig ovary. We could show the same thing at the protein level. And so you can see very clearly here in the thermoneutral pigs that there's no STAR. These are in prepubertal pigs, I should add. And in heat stress, that STAR, that catalyzes that first step in estrogen production, that really goes up. And the same thing is true about the enzyme that catalyzes the last step. So we know that there's changes to the ovary at the molecular level that would translate into an effect on the hormone environment within the animal. This pathway here, I'm not going to drive you crazy with a whole bunch of pathways, but this one in particular it seems to be the pathway that's very central within the ovary to drive kind of some of the effects that we're seeing. And so this is in heat stress pigs again. This is two components of that pathway, and you can see that in the heat stress, the black bars, that it's really elevated by heat stress. And this is just, it's kind of bright in here, but this is an ovary, section of an ovary from a thermoneutral pig, and this is from a heat stress pig. And really all I want you to kind of recognize is that green is higher in this ovary here. So it's shown that this pathway is activated as well in heat stressed animals. Now, why am I telling you that? Why do you need to care about that pathway? This is the pathway basically, and it's activated by insulin. I just told you that heat stressed animals become hyperinsulinemic. So that was shown in the liver about 20 years ago now, and we've actually shown that this pathway also in the ovary regulates how the ovary basically detoxifies chemicals. So we know that it's really a critical pathway in the ovary for the ovary being able to defend itself from a chemical exposure. And two of these, sorry, I should back up here. Two of these, I'm going to show you some data as we go along on these enzymes. They are involved in converting structures, chemical structures, to make them more easily excretable from the body. And we've studied them for quite a few years. There's very little known about them in the pig. Actually, very little is known about how the pig ovary actually detoxifies chemicals at all. So really, this is all new, right? This is all new information. So what we found, these were in postpubertal pigs that were just heat stressed for 5 days, so during the follicular phase, right up before ovulation. And the blue here is thermoneutral and the red is heat stressed. And so you can see this one protein, for example, was decreased by heat stress, whereas this one was increased. And this protein is involved in detoxification of many different chemicals. It's known for that role, right? So less of that during heat stress would make that heat stressed pig far more susceptible to chemicals which it's exposed. So this is kind of what we've been working on. So the theory being that heat stress, we know causes hyperinsulinemia. We know insulin regulates this pathway that's important for chemical biotransformation. So we next asked the question, is really a heat stress pig more sensitive to a chemical, right? So that's what we asked, that's the question. Does a heat stress pig respond differently to an environmental exposure than a thermoneutral pig? And so the chemical that we decided to investigate in this study was a mycotoxin, zearalenone, which acts like estrogen. It's important for the swine industry. Swine are super sensitive to it. This is just showing you that, that we're mentioning the derecho again, right? But there was an increased mycotoxin risk in Iowa. This is something I'm sure you know, and this is from Biomin, who did this survey of really this was the corn risk by state for zearalenone kind of contamination, and you can see we're pretty high. And so that's the chemical we chose. We wanted something that was relevant to the swine industry when we started doing this work. And so I'm showing you here the kind of experimental design. And PF stands for pair-fed, and these are control pigs. They got control. Pair-fed pigs who got zearalenone, heat-stressed pig who got a controlled diet, and heat-stressed pig who got zearalenone. And I want pair-fed, it's important that they're pair-fed because heat-stressed animals go off feed, they eat less. And so what we do is we stagger these experiments so we know that the pair-fed thermoneutral pigs are getting the same amount of feed as the heat-stress are, because we want to be able to eliminate it being a feed intake effect, right? We gave them zearalenone in cookie dough to make sure they got the exact amount that we wanted them to, and to make sure they ate it all, and they love cookie dough. They were all very excited when they heard you come with cookie dough. We tested the feed out at the swine nutrition farm here to make sure there was no zearalenone contamination. And so they were, so the animals were acclimated and brought in. We got a serum sample here, and then we gave them controlled diet or zearalenone within cookie dough for 7 days. And so the thermoneutral pigs were kept in about 20 degrees Celsius rooms, and the heat stress was about 35 degrees. And so we measure then their respiration rate and their core body temperature and all of that to make sure that they are experiencing heat stress. So what I'm showing you here is that this PC is the pair-fed control. So the thermoneutral animals who got the control diet, and these are the thermoneutral pair-fed animals who got the zearalenone. And then we have the heat-stressed who got control and heat-stressed zearalenone. We actually have a group of pigs too where they just ate as much as they wanted and they were in thermoneutral conditions. But I just didn't include them here just to keep it a bit more simple. All right, so serum estradiol, so that's the main estrogen in females, and so we measure that to see if there's an effect of zearalenone on estradiol, or on that main hormone being produced from the ovary. And you can see here that there's no effect of zearalenone in the thermoneutral animals on serum estradiol. Heat stress itself caused there to be less estradiol, so heat stress alone has this impact. And then in the heat-stressed animals who got zearalenone, they did have an increase in estradiol production, but really just brought it back to what the thermoneutral animals were. So there's an effect on estradiol by both heat stress as well as zearalenone in the heat-stressed animals, but not in the thermoneutral animals. This is to show you that zearalenone exposure in the thermoneutral reduced ovarian weight, but we didn't see this effect in the heat-stressed animals. We are going to keep going with this and count. We section up the whole ovary and we count each and every one of those eggs, and it takes a lot of time, but we're working on that right now. So again, a different effect between the thermoneutral animals and the heat-stressed animals, so kind of, you know, it kind of corroborates with our hypothesis that they'll respond differently. And then we measured vulva area, and so the vulva in pigs is really sensitive to estrogens. That's why the vulva gets swollen right before ovulation. It's a very estrogenically sensitive organ. And so what we show here is that there's no effect of zearalenone in the thermoneutral pigs on vulva area, but there is an effect in the heat-stressed animals, such that you have an increase in vulva area. So that might translate then into an animal appearing that they're in heat if they're an animal looking that they're ready to be inseminated, right, or they're right before ovulation when they actually weren't, right? But it's also shown that there's an estrogenic effect of zearalenone in the heat-stressed animals, but not in the thermoneutral. So again, a difference between those two, right? And then the last thing I'm going to show you to do in the heat-stressed animals is that we then look to see if there's an effect of heat stress and zearalenone, and then in combination, on the ovary's ability to detoxify chemicals. So looking at those 3 proteins that I showed you earlier in that little kind of schematic of the cell. And so, like I said before, there's really no information on how the ovary detoxifies chemicals in pigs, and so this data is quite new. And so you can see here one of the proteins is decreased by zearalenone exposure in the thermoneutral pigs, but it's actually the opposite effect in the heat stress. It's increased, right? So a chemical that this enzyme works on, you would see totally different effects based on whether the animal is in thermoneutral conditions or heat stress. This enzyme here is very much involved in detoxification of many, many different chemicals, and it's increased by zearalenone in both the in both of the pigs, regardless of whether they're in thermoneutral or heat stress conditions. It's increased. So it's responsive to zearalenone. It doesn't matter if the animal is hot or cold. And then, oh, excuse me, holy moly, what did I do? Then this last one here, this is another enzyme that's involved in detoxification mainly. And really there was no effect on zearalenone. So in any of the treatments where zearalenone is present, nothing happened. However, in heat stress alone, there was a dramatic increase in the abundance of this enzyme in the ovary. So what does that mean, right? It really, all I'm telling you is I don't know how these chemicals detoxify zearalenone specifically, or how these enzymes are doing it. We haven't studied the structural changes in zearalenone when they are acted upon by these enzymes. However, what I'm showing you is that heat stress really creates a different environment within the ovary to be able to deal with the chemical exposure. Okay, so I mentioned earlier that there's common metabolic changes in obesity and heat stress, and these are really 3 of those. Hyperinsulinemia. They also become systemically very inflamed, so there's inflammation all around the body. And then they have metabolic endotoxemia, and that really results from a leaky gut, and then whatever's in the gut leaks out into the system, and there's an inflammatory response. And so we know that these are present from many years of work from many people other than me at Iowa State. However, we also know that they're present in obesity. Obesity is really a case where very, very associated with female infertility, right? And so I'm not, trying to give you a whole spiel about human obesity, right? This is Swine Day, right? But I want you to think about, like, when you have an animal that has a higher, maybe more fat on the carcass, how could that actually be also kind of playing into what we see with chemical exposures? And I wanted to introduce this because I have a brief study I'm going to show you on these perfluorinated chemicals. So I kind of want to make the ploy that these are really important, and kind of like Dr. Holtkamp just talked about, there's something that we need to be thinking about being responsive to now, not in 10 years or not even in 2 years' time, when maybe there's more regulation of animal products that are contaminated. Okay, we've also shown in the ovary of obese females that they have this low level of kind of constant DNA damage. They're not able to fix their DNA damage. They have a problem. It just gets kind of broken. They have changes in how they can alter or how they can respond to chemicals. They have changes to their estrous cycle, and they've got altered levels of hormones. Very similar to what we see in heat stress pigs. We know that in obesity, so this is using a model this is a mouse model, and we know that in obesity, if we study when these mice get fat, they just overeat the same food as these wild-type mice. They lack the satiety signal. So it's like when I get a big bag of Cheetos, and I can't stop eating them, right? They just cannot recognize that they're full up. And so they gain weight. You're not giving them high fat, high sugar. You're just giving them the same diet. They gain weight, they become fat, they become hyperinsulinemic, and they've got all the metabolic features associated with obesity. And we know this graph here is showing you changes to that PI3 kinase signaling pathway over time in the obese animals. And so it goes way up, some components of it, and this component here isn't affected at all. But really, we wanted to study this because, well, we're interested in kind of a human aspect too. When females tend to start putting on weight, they generally don't lose it again. They kind of stay at that high body mass phenotype. And so that we wanted to study kind of the onset of obesity as well as it progresses. So we found that, yeah, not surprisingly, similar to the heat-stressed pigs, we find this impact on this pathway that's important. And we also then looked at those same 3 chemical biotransformation enzymes, and again, we find that there's dramatic effects depending on basically that body composition of the female, right? And so some of these would translate into more detoxification or hopefully more detoxification. But sometimes these enzymes can actually bioactivate chemicals too. Not many, but they do. But generally, it's kind of similar to that heat-stressed female, right? The environment within the ovary is completely changing. And it's changing because of systemic signals from the body. And so this is kind of the scheme here. We did the same thing, obesity, hyperinsulinemia, this pathway that's changed, and we looked at ovotoxicity. And really the reason I wanted to introduce this is because of this group of chemicals that I'm going to talk to you about now. So if you haven't heard about these chemicals, you should just Google forever chemicals, and there's a whole wealth of information out there, and they really are a concern. Like, I think they're one of the biggest concerns that we have right now. They're everywhere because they've been used for decades. All right, and so they're in anything that's flame retardant, fabric, like our chairs here, curtains, furniture. They're in all of that. They're used also in firefighting foams, and so water around army bases and airports and stuff tend to be quite highly contaminated. They're used in Teflon, so in non-stick cookware. They're used in food packaging, popcorn, the inside of popcorn bags. They're everywhere. I mean, they're literally everywhere. They're used in cosmetics also. So these are just some kind of recent, and I'm sorry, this one's quite small, but these are just some recent stories over the last couple of months. And the Department of Defense are really trying to figure out PFAS contamination across the country because we don't know how to remediate it, and we don't know how to fix it. This is from a few days ago here, and I'll show you why this is relevant in a second. But what was considered to be safe in our drinking water was dramatically reduced 2 weeks ago, and so it's not considered to be safe. And so we really have no clue what's in our swine drinking water. And so these are really persistent in the environment as well because they've got these carbon-fluorine bonds that are resistant to a degradation by bacteria in the soil, by heat, by sunlight. They really are very, very, very stable in the environment. Are we exposed 99% of the US population have PFAS that's measurable in their blood, in their urine, in their nails, any biospecimen. So if we measured all of us here and got a urine sample, we would almost for sure find PFAS. I should clip back 1 second. They're known as perfluoroalkyl and polyfluoroalkyl substances, or PFAS, and there's a whole bunch of them, a whole bunch of these chemicals within this kind of chemical class. So they're present in the human population. Are they in our animals? We don't know for sure. The half-life is something that should concern you. This PFAS and PFOA are two that have been used for the longest time. They're really legacy chemicals. They're very persistent in the environment. They have a half-life in the human body of years, right? So that means the amount it takes to get a chemical from a certain level to half, to lower it by 50%. Not to completely eliminate it, but just to lower it by 50%. So they've got years and years of persistence in the body. They actually, what I told you about biotransformation is completely irrelevant to these chemicals. They don't seem to get biotransformed. They wouldn't stay in our bodies otherwise, right, for such a long period of time. This is a map of Iowa from the DNR website. There's a dashboard that's updated regularly, like whenever they get new data, it's updated. And they are surveying water around the state of Iowa to get a handle on what our PFAS contamination is. So let me kind of orient you here. That's where we are here in the middle. If the circle is green, it means they've measured and not detected it. But if the circle is orange, they have measured and they have detected PFAS in the water. And this is detected below HA. That's the health advisory. And so that health advisory was 70, I think it's mgs per liter, until 2 weeks ago, right? So I think they're going to have to kind of revise whether they're above or below the health advisory. And you can click on any of these dots and see what the levels were. You can see exactly where they were measured, and you can click on and see what level it actually was present. But you can see that they're detectable around the state, so wherever there's an orange box, it's detectable in our water. Okay, I'm getting there. And this is from like an Ames City Council meeting on Tuesday. And so just to show you that this was the old EPA health advisory, 70 parts per trillion, and it's dropped dramatically since 2 weeks ago. And so now the Ames water is much above the new health advisory, right? And so this is the drinking water in the faucets in Ames, but we have no idea how it affects our swine. And I'm just going to show you a study where, in 1 second, and I'll be super quick, and it's not to give you all the details, but just to make the point. And if this is all I get through, this is fine too. This is kind of showing you a couple of cases. This is from June of 22, like just a few weeks ago, and this is in New Mexico. And this farm is downstream of a large contaminated site, and so they had to euthanize about 4,000 cows. So it's becoming a big problem for producers. This is another story from like last week, a week ago, about farmers that have contamination of PFAS in Maine. And I want to draw your attention to this. For many of those farmers, it means their crops, meat, eggs, and milk are now unsafe to eat. So I think this is a big problem, right? And we don't really have a handle on it yet. You can kind of look at this later if you want, but there's a good few reviews out there, very few dealing with livestock and wildlife, but this one does. And this is kind of a schematic showing you like all the places PFAS can come from. But what's interesting here, this table is showing you the half-life of those two legacy chemicals, PFAS and PFOA, across different species. And so we see here pig, and this is in a female pig, 1.7 years for PFAS and almost a year for PFOA. Again, that's the half-life, the time it takes for these chemicals to be eliminated by 50% from the body. So they're detectable in our animals and probably should be a concern for us. There's a lot of evidence out there that they are reproductive toxicants, so they're associated with many reproductive effects in humans. And then there have been some animal studies lately coming out showing that the estrous cycle is being altered, earlier puberty onset, loss of those very immature eggs within the ovary, alterations to reproductive hormones. So there's evidence out there that they have a reproductive effect. And I'm not going to go too much into this detail, but we did a study in lean and obese mice to figure out how they're affecting ovarian function. Gave them 14 days, we gave them just saline or PFOA by mouth, and we did a whole bunch of assessments. This is really, there was no effect on PFOA on body weight. The obese ones were just heavier. There was no effect of PFOA on estrus cyclicity. There was an effect on liver weight. Almost always in every study where these chemicals are exposed to an animal, the liver weight almost doubles. And that's a big concern, right, for animals' health. And ovary weight was also decreased in the PFOA-exposed lean mice. And we looked at, did all this work to figure out what proteins are changed within the ovary, and I can tell you that they are. And not only are they changed, but they're different in lean and obese mice. So some proteins consistent, some are different. These are ones that we're working on now within our lab that are very consistently changed, but they change in different directions. But we have this one, for example, is we know it has an important reproductive role, so we're following up on that. So this is my last slide. We know that heat stress causes a whole bunch of reproductive effects in pigs, and we know that there seems to be a combination. There's effects of chemicals on pig reproduction, and there seems to be an additive effect of heat stress also. And then this are the take-home messages. Systemic endocrine disruption messes up steroidogenesis and ovarian function. And there's different effects of these chemicals on the ovary if an animal is heat stressed or obese. PFAS chemicals are a risk that we need to be getting a handle on. And we know that ovotoxicity doesn't just depend on toxicant exposure. I mean, it kind of does, right? The more of a toxicant you get, the worse it probably is. But it also depends on metabolic status of the animal. So with heat stress and maybe altered BMI of animals, that's something to think about. And these are the guys who've done all the work, and I really want to acknowledge the Iowa pork producers because they've given us most of the money to do this work in swine. So leave it there. Questions?
One question.
Yeah.
Did you examine the livers?
Yes.
If there was fatty liver and
Yes.
there was position?
Yeah, there is. So PFAS are very associated with fatty liver, and we have done work on looking at gene expression in the liver, and even looking at them, like they look kind of yellowy and slimy. So yeah, we have. I think that's a problem, right? Because liver metabolizes everything, including our steroid hormones, and so I think that the liver and the ovary, though, that connection is an issue too. So we're following up on it, but yeah, we haven't done that in pigs. In mice, we did it. Yeah.
So heat stress of ovaries, we know from the time the sow girls, she's beginning to have form eggs for the next litter.
Mm-hmm.
Is there a time from that point there, when is heat stress the worst? The following litter performance?
Yeah.
Is that an open question?
Yeah, no, that's something we don't know. So there's a time in utero when the ovary is developing in those piglets. There is a stage. So I think that's critical for their formation and endowment of the ovary, what's there to begin with. We haven't studied it, so I don't think anyone really knows if heat stress is affecting it. However, then I think afterwards, after puberty, when they're cycling and so on, if you clear out that pool faster, and the time frame of seasonal infertility and how long it lasts would indicate to me that it is having an effect on those immature follicles, because the time, they take about 70 days to get to ovulation status. And so I think that kind of implies that there might be an effect too on those very immature follicles. Yeah, because if she's having problems, yeah, that could have impacts. But we don't, nobody's really studied on that immature ovary that's developing in utero, but I think it's ripe for studying. And I know Jason Ross and Lance Baumgart, they did a study a while back with heat stress during gestation. They didn't look at the ovary, but they looked at the sensitivity, I think, of those animals to heat stress in later life, and they also found that there was a higher fat composition on the carcasses, depending on whether the piglets were exposed to heat stress in utero or not, and the stage of gestation mattered. So, yeah, good question.
We have time for one more question. There's one.
Don't be shy, don't be shy.
If not, let's give. There is a question.
Yeah.
Sorry.
Yeah, that's a good question. So there are, like, not all the endpoints react the same way. Even in obesity as well, we've done a bunch of work with obese. So with the ovary weight is one in particular, where in the lean animals, there was an effect of the thermoneutral, there was an effect on zearalenone, but not in the heat stress. We've seen the same thing in obese females too, given chemicals. They don't seem to be able to clear out the damaged eggs as well as the non-metabolically stressed females. There seems to be a defect in getting rid of bad quality eggs. And so we've kind of identified some pathways that are involved in that, but that's what I think is going on. I think it's not that they're not being damaged, the follicles, I just don't think they're being cleared out. And that could be a problem too if they're fertilized, right, if they're poor quality oocytes. So that's something we've been trying to get at for the last while also.
If there are no other questions, let's please give Dr. Keating another round of
Thank you.
applause.